Dear friends and my dear students, I am Dr. P.K. Verma. Today, in the third session of Human Physiology, I shall discuss digestion and absorption of food. Our food mainly constitutes of protein, carbohydrate, and lipids, along with a small amount of nucleic acids, vitamins, and minerals. These complex organic molecules, that is protein, carbohydrate, and lipids, cannot be directly absorbed by mouth and alimentary canal. Therefore, we need a digestive system by which the food components are digested, absorbed, and assimilated. We have got a very efficient digestive system comprising mouth, alimentary canal, and digestive glands like salivary glands, gastric glands, intestinal glands, liver, and pancreas. Now, what is the digestion? Digestion is the process of enzymatic hydrolysis of complex organic foods into simple, soluble, diffusible, and absorbable constituents. Now come to the first gland, salivary glands. In the oral cavity of humans, there are three pairs of salivary glands. These are submandibular, sublingual, and parotid glands. The glandular cells form a single layer around a large space called acinus or alveolus into which secretion is stored. A number of small intercalated ducts from the adjacent acini join together to form larger ducts that finally open into mouth. In man, about 500 milliliter saliva is produced per day, but the volume may increase to 1500 ml, in which 70% is contributed by submandibular glands, 25% by parotid gland, and only 5% by the sublingual glands. The taste buds on tongue become stimulated when some soluble and sapid substance is actually present in solution. When food comes into mouth, salivation starts and the saliva contains mucus secretion to lubricate the food, and the serous secretion contains enzymes alpha amylase or tyline and lysozyme. Amylase is activated at pH 6.8 in presence of chloride ions. The pH is maintained by bicarbonate, phosphate, as well as mucin. Amylase digests alpha 1,4 glycoside bonds present in starch, that is amylose and amylopectin, and the glycogen, but not the beta bonds of cellulose. So, all polysaccharides cannot be digested by alpha amylase or salivary amylase. By acting at random locations along the starch chain, alpha amylase breaks down long chain polysaccharides ultimately yielding maltose from amylose and also limit dextrin from amylopectin. Alpha amylase cannot act near branching points, so a tetrasaccharide dextrin also is formed in a small amount. The lysozyme can digest bacterial cell wall and kill them, so prevent their infections. At pH 7.0, saliva is saturated with calcium, so that teeth do not lose calcium, but at pH 5.5, calcium is lost rapidly. Therefore, the acidic food can damage your teeth. In the mouth, therefore, almost neutral pH should be maintained. Now, what is deglutition? After the food is masticated well, it is swallowed. Whether it is the solid food or a liquid, the process of swallowing, also known as deglutition, is same. The process involves three activities: buccal, pharyngeal, and esophageal activities. The mouth is closed and the bolus collected on the upper surface of tongue is forced into oropharynx by an upward and backward movement of tongue. At the same time, the soft palate rises up and approaches the posterior pharyngeal wall, which is brought forward to close the nasopharynx. Uvula also spreads laterally. The inferior constrictor muscles of pharynx then closes firmly and prevents air to enter into esophagus. The larynx begins to rise as the bolus passes over the back of tongue. The tongue moves back towards the posterior pharyngeal wall like a piston and forces the lobus towards pharynx. The epiglottis becomes folded to form a cowl-like hood over the pharyngeal orifice. Glottis is closed by action of sphincter surrounding it. The hood passes over the lateral edge of epiglottis in two streams into the part of the pharynx posterior to larynx and then into esophagus. The sphincter of esophagus opens for a second and the bolus passes down. The larynx drops to its original position, vocal folds open, and the epiglottis quickly resumes its original position. Thus, at the time of swallowing, breathing stops for a while, and once the bolus moves into esophagus, breathing can again start. Now, there is no gland in esophagus except mucus secretion from its wall. However, in the stomach there are gastric glands. In the pits between gastric villi, known as gastric pits, the bulbotubular or tubulo-alveolar glands or gastric glands containing peptic cells and oxyntic cells are present. Oxyntic cells are also known as parietal cells. These cells secrete hydrochloric acid. The secretion of hydrochloric acid is under direct control of a peptide hormone gastrin released from the wall of pyloric stomach after arrival of food. Human gastrin 1 has molecular weight 2114. It has the composition glutamic acid, glycine, proline, tryptophan, leucine, then 5 molecules of glutamic acid, alanine, tyrosine, glycine, tryptophan, methionine, aspartic acid and phenylalanine. The last 4 amino acids, tetrapeptide, act as active center of the hormone. In gastrin 2, tryptophan is sulfated. Hydrochloric acid is secreted also due to vagus impulse. The secretion is also affected by gastrin released from delta cells of islets of Langerhans. The vagus nerve terminals present in the wall of the stomach initiate gastric secretion as well as peristalsis. See this figure. Brain has the center of control of all metabolism. There is a vagal center in the medulla. It is associated with 10th cranial nerve and the branches innervate the wall of stomach. The vagus impulse as well as the hormone gastrin control the secretion of gastric glands. Now you can see the parasympathetic site, pepsin and acid production, gastric phase, that is local nervous secretory reflexes, vagal reflexes and gastrin histamine stimulation initiate gastric secretion, and finally the intestinal phase, nervous mechanism and hormonal mechanism take place. From parietal cell, proton is produced by breakdown of water. It is transported into lumen of stomach by active transport system known as proton pump. The proton is followed passively by chloride. Peptic cells are also known as chief cells or zymogen cells. They secrete zymogen granules containing propepsin or pepsinogen. In the acidic medium, that is between pH 1.4 to 4.5, the enzyme pepsin is activated and causes hydrolysis of insoluble polypeptides into smaller fragments or oligopeptides. These semi-digested proteins are also known as peptones and proteases. Pepsin breaks the peptide bond with tyrosine or phenylalanine. Now, in the stomach, when food comes, a wave-like movement starts that is known as peristalsis. By this movement, food is mixed well with acid and enzyme as well as moves backwards. This movement is under control of vagus nerves. The gastric motility is greater in its lower part of towards pylorus. The motility decreases when some acidic materials pass into duodenum or emulsified fat is present in it. Chemicals like atropine and metoclopramide also decrease peristaltic activity. This diagram shows how anteriorly the wall of esophagus, stomach or intestine contract, but the posterior walls relax so that bolus is pushed backward. They move gradually from stomach to duodenum and then into ileum. The same is shown with the help of the complex system of nerves and the sensory terminals present in the wall of stomach. Sometimes the direction of wave changes and the food moves forward. It is called antiperistalsis or retroperistalsis. It results into vomiting. It is an involuntary function. Local irritation of stomach, such as bacteria or food poisoning, activates the emetic center of the brain that in turn signals an imminent vomiting reflex. Food then moves in the opposite direction, often from duodenum into stomach. Now, by a normal peristalsis, gastric evacuation takes place. In the stomach, 4 to 5 hours are needed to convert food into a semi-digested and paste-like mixture commonly known as chyme. The pyloric sphincter opens gradually and chyme passes into duodenum bit by bit. Chyme stimulates duodenal mucosa to release certain peptide hormones. Hormone secretin released from duodenal mucosa controls secretion of pancreatic juice, and cholecystokinin controls secretion of bile. In mammals, there is a third hormone, pancreogymin. It has dual activity. That is, it can act on gallbladder as well as on pancreas. From the wall of upper part of ileum, another hormone, enterogastrone, is released that inhibits gastric gland and prevents its secretion. From the wall of middle and lower part of ileum, one more hormone, villikinin, is secreted that increases movement of intestinal villi. See in this photograph, liver, gallbladder and pancreas have been shown. The pancreas is present within the loop of duodenum, but a longer part or the head and body part of pancreas lies on the surface of stomach. Gallbladder secretes bile. Bile contains 89% water, 11% solid materials. Among the solid, bile salts comprise 6%, cholesterol 2.5%, phospholipids 0.4%, inorganic salt 0.8%, and the pH ranges 7 to 7.6. When chyme comes into duodenum, from the mucosa layer of duodenum, cholecystokinin is released. Cholecystokinin causes contraction in the wall of gallbladder and opens the valve of Oddi, so the bile is released into duodenum. In mammals, one more peptide hormone, pancreogymin, is released that can cause release of bile as well as pancreatic juice. When chyme mixes with bile and due to presence of high concentration of bicarbonate, it is neutralized and the pH gradually rises to 7.6, at which the pancreatic enzymes can act well. Bile contains bile salts, glycocolate and taurocolate. These are highly soluble and act as powerful detergents. When they mix with fat and oil, they cause emulsification and dissolution of fat molecules. When these molecules are dispersed well, they can be acted upon by salivary lipase and digested well. In absence of these salts, such as after surgical removal of gallbladder, therefore digestion of fat is highly affected. Now see a better view of duodenum and attachment of the pancreatic duct and common bile duct to the loop of duodenum. The opening provided with valve of Oddi also have been shown. This is the transverse section of pancreas, the bunch of cells. You can see these are acinar cells and connective tissue is clearly seen. When thoroughly searched within connective tissue, you can find these islets of Langerhans as well. Pancreas is a compound gland that has about 60 to 62% part made of enzyme secreting exocrine cells known as acinar cells. The acinar cells form small follicles at the tip of fine pancreatic ducts. These cells release proenzymes of different digestive enzymes into the follicles and carried away by the ducts. All such ducts join together and open into duodenum by a common pancreatic duct near the ampulla of common bile duct. The pancreatic enzymes are released under control of duodenal peptide hormone secretin as well as pancreogymin. The mixture of enzymes is commonly known as pancreatic juice. Pancreatic juice contains alpha amylase, lipase trypsinogen, chymotrypsinogen, and aminopeptide, along with a small amount of nuclease and elastase. There are intestinal glands. These are tubular in feature. The glands present in duodenum are known as Brunner's gland and they secrete only mucus. But in ileum, there are several crypts of Leverkuhn that secrete intestinal juice or succus entericus containing carboxypeptidase, disaccharidases, and a little amount of lipase and nuclease also. Now come to the process of digestion of carbohydrate. Carbohydrate digestion begins in the buccal cavity. Saliva contains large amount of alpha amylase, commonly known as ptyalin. Ptyalin acts on polysaccharides, starch and glycogen containing alpha 1,4 glycoside bond and at places alpha 1,6 glycoside bonds. Enzyme breaks alpha 1,4 glycoside bonds at alternate positions, so a large number of disaccharide maltose can be formed. However, it cannot hydrolyze glucose around the branching point, so some tetrasaccharide molecules known as limit dextrins also are formed. Unfortunately, in animal system, alimentary canal has no debranching enzyme, so the branched limited dextrins are ingested with fecus. When the food comes into a small intestine, salivary amylase gradually becomes inactive in the stomach, but again the pancreatic amylase becomes active. The mechanism of action is same, that is salivary amylase as well as pancreatic amylase digest polysaccharides into maltose and limit dextrin. These amylases cannot digest cellulose that has beta 1,4 glycoside bonds. When chyme comes into duodenum and is neutralized by bicarbonates of bile, starch and glycogen can further be digested by pancreatic amylase that acts on mild alkaline pH, that is 7.6. So salivary amylase acts at pH 6.8, whereas pancreatic amylase acts at 7.6. In our food, other disaccharides like sucrose or invertose and lactose also are present in a small amount. These are digested finally by the enzymes present in intestinal juice. Enzyme maltase. It digests maltose into glucose and another molecule of glucose. Enzyme sucrase digests sucrose into glucose and fructose. And finally, enzyme lactase digests the milk sugar lactose into galactose and glucose. So, with the help of these intestinal enzymes, all disaccharides are converted into monosaccharides. Of course, glucose is the major product of carbohydrate digestion. Fructose is present in a smaller quantity and galactose in very small amount because it is found only in the milk and the milk products. You can see this diagram. Pancreas is shown to be attached in duodenum. The pancreatic amylase is poured into duodenum. Carbohydrate digestion begins, producing maltose, dextrins, and smaller oligosaccharides. Further digestion takes place by intestinal enzyme. Maltose forms 2-glucose, sucrose forms glucose and fructose, and lactose forms glucose and galactose. Alpha D glucose is most abundant sugar in our food, but we can get galactose from only the milk or milk products. Fructose is present in all plant resources. The common sugar, sucrose or invertose, is major source of fructose. Now there is a problem known as lactose intolerance. Many persons, particularly the adults, fall ill when they take milk. It is because they have no lactase enzyme at the brush border of a small intestine, so they cannot digest milk at all. In such persons, indigestion takes place after taking milk. Lactose intolerance is prevalent in blacks, Mexican American almost 65%, as well as in Oriental region 5 to 15%. When food comes into large intestine, it has only the food residues that were not digested and absorbed in the small intestine. In the vermiform appendix and cecum, herbivorous animals there have a large population of bacterial species that synthesizes cellulase enzyme. This enzyme cellulase can break cellulose into straight chain polymers, that is beta-D glucose. But the end product is not beta-D glucose, rather it is beta-D gluconic acid. So it is not a process of hydrolysis, rather oxidative decomposition. However, it is common saying that in the appendix and cecum, cellulose is digested. Now let us study digestion of protein. Protein digestion is most complex component of our food and also variable in nature. Most food contains large chain called polypeptides, which are often insoluble in water. To digest various types of proteins, we have large number of protein digesting enzymes. When protein comes into a stomach, it itself can stimulate release of gastrin hormone from the wall of a stomach that controls gastric secretion. Hydrochloric acid on the one hand kills germs, bacteria, etc. present in food, dissolves many hard parts of food, being itself a hydrolytic acid, and converts propepsin into pepsin. Pepsin is an endopeptidase enzyme that breaks insoluble polypeptides into oligopeptides or smaller fragments of protein commonly known as peptones and proteases. Pepsin can act very poorly on the soluble milk protein casinogen, which is the major constituent of infants' diet. Therefore, in mammalian children, a special enzyme renin is secreted that causes curdling of milk very fast. Renin causes precipitation of milk protein into calcium caseinate. Now, after their solidification, they can be digested by pepsin. In adults, renin is absent, so milk protein is poorly digested. In the small intestine, large amount of trypsinogen and chymotrypsinogen are released with pancreatic juice. Trypsinogen at first is activated into trypsin by action of enzyme enterokinase released by intestinal mucosa. Once a small amount of trypsin is formed, it catalyzes formation of other molecules of trypsin and chymotrypsin and activates other protein digesting enzymes. Like pepsin, trypsin and chymotrypsin also are endopeptidases and hydrolyze polypeptides into oligopeptides, but these enzymes act in alkaline medium rather than acidic medium. Carnivorous animals have another enzyme, elastase, that digests elastin fibers present in fibrous connective tissue. Now, in the first line, you see trypsin is activated by enteropeptidase and form trypsin. Then all molecules of trypsinogen, chymotrypsinogen, pro-carboxypeptidase, pro-elastase, etc. are activated by trypsin itself into the active enzyme chymotrypsin, carboxypeptidase A and B, elastase, etc. The oligopeptides are finally digested by two exopeptidase enzymes, the carboxypeptidase and aminopeptidase. The former is released from pancreatic juice and later with intestinal juice. These enzymes act at carboxyl and amino terminals of oligopeptides and release the amino acids one by one. In this way, digestion of protein is completed in the posterior part of ileum. Now lastly, the lipid is digested in a small amount. The bile salt, glycocolate and taurocolate cause emulsification and dissolution of fat. However, they have a limited concentration in bile, so hardly 10 gram fat can be digested every day in us. The remaining fat passes out with fecus. If we take large amount of fat, digestion is affected. In our alimentary canal, only the smaller glycerides, that is fat and oils, are digested, not all the lipids. Lipase acts on the ester bonds and release fatty acid from glycerol. However, we cannot digest much of the dietary fat because of lower amount of the enzyme as well as the protein as well as the problem of emulsification. The steroids and fat soluble vitamins are absorbed without being digested, but the triglycerides and phospholipids are digested by pancreatic lipase. This enzyme is released also from the intestinal wall in a small amount. When a phospholipid is digested, it contains two fatty acids and the glycerol phosphate separates from fatty acids. Phosphate is later on released by alkaline phosphatase from the glycerol molecule. However, glycerol phosphate itself can pass through the intestinal wall into lymphatic vessels. Now, when digestion is completed, the time comes for their absorption. All the amino acids and monosaccharides formed in a small intestine are absorbed partly by diffusion and partly by active transport. These are taken up by blood and carried to liver by hepatic portal veins. A major part of glucose is retained by liver and converted into glycogen. But amino acid pass into body for utilization elsewhere. Glycerol and fatty acids are absorbed and pass into lymphatic nodes known as Peyer's patches and the lymphatic vessels. Here they again become triglycerides in the form of small droplets known as chylomicrons. They move into adipose tissue where most of them are stored for future use. In the large intestine, mainly in the cecum and ascending colon, 350 ml water is absorbed when 500 ml undigested food residues passes from a small intestine. Sodium chloride, some amino acids and certain anesthetic drugs, aspirin and some steroids also are well absorbed, but protein, fat and sugar as well as calcium are not absorbed in large intestine. In the residues commonly known as stool or fecus, some bile pigments such as stercobilin are present, which gives the characteristic yellowish color. If it is absent, the color becomes like clay. Fecus has large amount of plant products, indole and escatol, that produces bad odor. The odor is also due to gaseous products of the bacterial activity. In this figure, all parts of digestive system and their function have been mentioned. In the mouth, mechanical digestion, that is chewing and swallowing, occurs, and chemical digestion of starch into maltose takes place. In the stomach, mechanical digestion by peristalsis and chemical digestion of proteins by pepsin takes place. Lipid-soluble substances such as vitamins and a medicine like aspirin can be absorbed in the stomach. In a small intestine, mechanical digestion and propulsion primarily by segmentation by peristalsis and chemical digestion by enzymes takes place. The digestion products, the sugar, amino acids, fructose, lipid, water, minerals and vitamins take place in the small intestine. In large intestine, no chemical digestion except by bacteria takes place. Absorption of ions, water, minerals vitamins, and small organic molecules produced by bacteria. Now the last process is defecation, that is discharge of stool. A massive peristaltic wave in the transverse and descending colon drive the food residues onward into rectum, and the subject experiences a sensation of fullness, that is pressure, in the rectum and a desire to defecate. When the large intestine, colon and rectum become full due to their pressure, vagus receptors are stimulated. It controls contraction in the part of colon and rectum, closes intestinal sphincter and looses anal sphincter. Defecation is a complex process involving not only these muscles of large intestine but also contraction of diaphragm and the skeletal muscles of abdominal wall. The levator ani muscle in the pelvic floor also aid defecation by pulling up the anal canal over the fecal mass. During defecation, the pressure has important effect on the circulation, especially on the venous return to the heart. Now you know everything related to your digestive system. In due course, you will also study disorders of alimentary canal. See you in the next session. Thank you. Thank you very much.